Hello, and welcome to our participants. This is an interesting time for the iPSC space. The science itself is very much emerging, but as we've learned from other modalities in the cell and gene therapy space, it is important to think about manufacturing early to avoid complications down the line. So in this panel, we'll discuss issues relating to manufacturing of iPS cell-derived cell therapies. And I'm joined by Dr. Greg Rusati, Interim CEO of Century Therapeutics, Dr. Stefan Braun, CTO of Cellistic, and Frederic Deguit, CEO of TreeFrog Therapeutics. Thank you all for joining us today. And to start off, I invite each of our participants to briefly introduce themselves and their companies, including the type of cell therapy products that they are making. And let's start with Stefan and then go to Frederic and Greg. Stefan, I think you're muted still.
Thank you. Pleasure also to be here, and thanks for the organizers for creating this great conference. So my name is Stefan Braun, trained as a stem cell biologist, 20 years of experience with human embryonic stem cells and iPS cells, founder and CTO of Encardia and Cellistic. Cellistic is our new company, and what we're doing with Cellistic is creating immune cell manufacturing processes, so very focused on T cells, NK cells, macrophages, and actually we bring that to the industry as a CDMO. So we partner with therapeutic developers that insert their gene edits in our manufacturing processes to create novel therapeutics, and we aim to bring them to the clinic, to the patients faster.
I'm Frederic Deguit. I'm the CEO at TreeFrog, and TreeFrog is, as you can hear, a French cell therapy company. We are the backbone of the company is a technology encapsulation of cells during the manufacturing process, not for implantation in the body. And we started working on the iPSC encapsulation and differentiation in the capsule. So we started on regenerative medicine and now moving to the oncology field with partners. And we also started to encapsulate now also mature cells, NK, T, MSCs, recreating a biomimetic environment for the cells in a capsule of a few hundred microns of diameter. So a new approach to manufacturing, and happy to share our views at TreeFrog with my colleagues here. Thank you, Gula.
Hi, Greg Rusati, Interim CEO of Century Therapeutics. Century is a product developer in the immuno-oncology space. We are using iPSCs to create T cells and NKs currently, and we have one product in the clinic, an iNK, an iPSC-derived NK cell in the B-cell malignancy indication. And we are also developing other NK products and gamma delta T cells for various indications in both hematology and solid tumors. We believe our product in the clinic right now is the product with the most edits, has 6 edits, and we have other products coming with more edits. So we believe our gene editing technology, our iPSC technology, our ability to differentiate iPSCs into NK and T cells, and our manufacturing and in-house manufacturing, also manufacturing capabilities are our strengths. So I have been the Chief Technology Officer of Century for about 3.5 years, leading those technical development manufacturing quality groups. And just my background real quick, I've been in cell therapy since 2006, was at Celgene prior to Century, did placental allogeneic as well as cord blood derived NK, and then did autologous CAR T, both at Beckman and Breyanzi. And before that, I was at Merck in biologics and vaccines.
Great. So in terms of the technology, people in the biotech space are probably most familiar with autologous cell therapies or those derived from allogeneic donor cells. So what are some key differences for iPSC-derived manufacturing that we should be aware of as folks think about this space? And maybe I'll start with Frederic, then go to Greg, and then Stefan.
Thank you. Well, that's a very broad question, but maybe because that's one of the focus of today with our partners is around the the integrity of the cells. iPSCs are very interesting cells because you can manipulate them forever somehow. But in real life, pluripotent stem cells they have a very short, let's say, appearance in the development of the embryo. It's a few days, and then they start to specialize into less pluripotent stem cells. And so in the lab, we tend to keep them for years and to amplify them for generations. And what we see is that depending on the technology you're using, you might end up with some difficulties to keep the phenotype of the cells. So the quality of the cell, what we call the quality of the cell, which is different from, let's say, the quality of the product, but the quality of the cells themselves, when you amplify them before you start the differentiation, I think is one of the key elements that is very different from amplifying a T cell or NK cells. And also, again, because we're coming from regenerative medicine, we know that the quality of the cells at the end will be key, because cells should remain there for some time, at least in the patient for some time. So I think we're taking more, let's say, more attention to this criteria of cell quality overall. But just an introduction to the point, because it's a very broad point. But
yeah, just to piggyback on that, I think it is the proliferative potential of the iPSCs that gives them the strength and many advantages. So because we can expand these cells quite a bit, it allows us to do multiple edits. That's something that you can't do with autologous, and you can't even do with primary derived allogeneic. So we have the chance to edit them more, put on more features, hopefully, which will help with cell persistence. And of course, you have CAR and other knockouts to avoid rejection. But we have other things that we believe will help with persistence, as well as maybe targeting solid tumors. So the additional edits that you can do because of the extra proliferative potential, because you are going to use up cell doubling when you do editing, of course, as you edit and sort, etc. And then the others are that we believe the ability to grow these cells indefinitely means we can make very large master cell banks. So one master cell bank per product, so one donor per product, which avoids donor-to-donor variability. That is going to be the case for donor-derived allogeneic. And then lastly, the scale we can get to, because we can make unlimited numbers of cells from a master cell bank, we can make batches as large as we want, whereas in primary cell-derived allogeneic, you're going to be limited by the number of doses you can make from one donor. So lots of advantages due to that unlimited proliferative potential. And I guess one last thing is, because you're using one bank, any effects of those edits can be thoroughly investigated and then controlled, and will be more consistent. Whereas if you're going to have to do it over and over with a new donor each time, it's going to be really hard to analyze and much harder to control from donor to donor what's happening with those edits.
Stefan?
I think key points have been mentioned by Frederick and Greg. So just one point that I just want to highlight. So Frederick is talking about the quality of the cells. Greg is talking about the master cell bank for the lifetime of the product. And so I think that is actually really critical. So we are doing these edits in iPSC, and we want to do them once, expand those cells, and then go into this complex process of manufacturing the cells to one of the target cells. So the iPS cell can still become all 220 plus cell types in the human body. And these manufacturing processes are a little bit more unique and special and complex than everything that cell therapy has seen before. So what is really critical is that a therapeutic developer understands that manufacturing process and understands what it takes to drive the cell to the target cell. And typically these processes are a little bit longer than what you see in an autologous process, because you have to go through all those development pathways to get to the right cell. So it's really critical to one, understand the quality of the cell, but two, to develop that scalable, robust, and reproducible manufacturing process wherein you instruct the cell to become the right cell type. So it's a lot of fun to bring all these technologies together and to work on a completely new modality.
Great. So actually, to follow up on that a little bit, as you just mentioned, there's parts of the genome engineering components that can be done just once to develop the master cell bank, and then you have the whole other part of the manufacturing process that is done for each batch with the differentiation, et cetera. So can you guys talk about what that means for GMP materials and analytics for those early steps that you go through just once to generate the master cell bank, and how do you characterize the genetic engineering that you do at that step? And then we can talk more about the kind of second half, the manufacturing of the individual batches afterwards.
I can start if you like. Yeah, so I think when you think about GMPs, I think a lot of people think it's all or nothing. It's not all or nothing. It's having the right controls at the right times for the right reasons. So when you think about autologous or even donor-derived, where you have to do the editing multiple times, your editing components, so the case of autologous, people use lentiviral vector, for example, a lot. So the plasmids that are used to make the lentiviral vector itself, you're going to have multiple batches of those because you're going to use them multiple times. Therefore, the bar is going to be a lot higher. You need to validate that process where you make your LVV, ultimately validate it for a BLA, and probably even have GMP vector for a phase 1 trial, probably. I mean, it could be negotiation. But you are going to have a pretty high bar to make sure you're making the same thing every time. So your testing's got to be validated. The process has got to be validated. When you do it once, it doesn't mean you have no GMP controls, but you don't need to prove that it's a validated process because you're only doing it once. You need analytical methods because you need to make sure what you're making is really what you made, but maybe those methods don't need to be validated. And then when you think about other controls, you need to know sourcing. You need to know whether the sourcing brings any risk, whether any animal source components, you need to test appropriately. So if anybody says one requires GMP, one does not, I think they're oversimplifying. I think one requires extensive GMP, and maybe iPSC requires suitable GMP controls that negate the risk that those types of editing steps bring. Not negate, minimize, probably better word, sorry.
We share that vision, using appropriate controls or phase appropriate controls. So the editing that iPSC developers typically do is using precision genome engineering. So understanding exactly which genes are edited at which loci, which genes are brought in again. And so that allows to do full genome sequencing, really understand what's happening with that cell, and that brings a level of quality. We believe that it doesn't mean that you have to do that full blown in a grade A clean room environment, because it might actually take you 12 months to fully engineer that cell line, maybe even 18 months. So there are actually paradigms that biologic developers used to do cell engineering, cell banking in controlled facilities, but not necessarily full-blown GMP. That the field is now adopting, and that we believe is appropriate.
If I could just add to that, Stefan, if you don't mind, I think it's a great analogy, because you think about biologic cell lines, right? Or I'll use a really more extreme case: live virus vaccines. Live virus vaccines mean use a live virus, which you're going to inject in people. It's going to be GMP grade, but that virus was derived from some virus early on, isolated from a human somewhere, not in a GMP environment, they isolated from a human. And then there was a bunch of lab studies where they, let's say, attenuated that virus. They played around with different ways to make that work. Non-GMP setting, not a clean room, for example. Maybe not even great, well, not even documentation, yes, right? You have to have good lab notebooks, et cetera, know what you did, but not like pre-written GMP batch records. So same thing here. You know, when you think about the editing you do, this editing is really complex. You don't know exactly the order of the steps it's going to work. You don't exactly know the promoters that are going to work. You play around. So the research groups have to have really good documentation so we know exactly what they use, because ultimately some of these cells are going to be the ancestors of the cells that go into man. So you need to know how you treated those cells. But having batch records to dictate everything they do would be ludicrous, because it's research. You don't know exactly how they're going to go. It's going to be meander, and meander might be a bad word, because it makes it sound like you don't know what you're doing. But it is going to be research. Things are going to work. Things aren't going to work. Try multiple things, multiple paths at once. So it's all about good documentation, but not the rigor of a clean room, like you said, Stefan, or the rigor of a batch record that says you must do things this way, because well, you could have some batch records and there's maybe no downside to that, but bottom line is good documentation, so you know what you did and where things came from.
I have nothing to add, Gula, on that, because we are not yet there. So we are building the platform to do our own gene editing, but we have not communicated on that yet. So I have nothing to add. Maybe just one comment on the situation, because we're working with a lot of partners. We are working on human material, so no cell is perfect. So no one is perfect. So when we look at gene editing in a cell that we know is not perfect, if you look really deep, you will find imperfections. So it's a question also of risk and risk mitigation, and more than anything else. So there is a limit there is no limit to what you can measure, but there is a limit to what you can let's say address as a problem ahead of testing your cell lines and testing the purpose of your cell line.
So on the topic of risk mitigation, actually, what kind of characterization do regulators want to see for iPSC-derived product, and what kind of issues do they tend to be worried about?
Well, the first thing that the field is faced with is the fact that a pluripotent stem cell, an iPSC is a pluripotent stem cell, can form teratomas. So it is a quote-unquote tumorigenic cell. If you put it in a kidney capsule or in a testis, a hallmark of that cell is actually that it will start to differentiate towards different types of cells. So undifferentiated cell types in the final product is not great, and so no therapeutic developer or regulator wants to put that in patients. So that is the first thing that people look at from a developmental biology perspective. So knowing what we put in these differentiation media, so the amounts of signaling pathways that we turn on and turn off is totally not consistent with pluripotency. So I think it's actually going to be really, really tough to have pluripotent stem cells still in a drug product. But it's obviously something that therapeutic developers need to check for. And luckily, there are very sensitive methods right now. So that's the first level of controlling. And here I give it to my panelists.
I think you're right. I think that there have been attempts in the past to implant directly iPSCs, but nowadays, no. Either you go down the route to let's say potent cells, but which are not pluripotent. I mean, maybe a checkpoint in a differentiation line, so you have a good control of the purity of your cell population and the fate of the cells. So you might and you see that, for instance, in Parkinson's disease, and people inject progenitors of brain cells. So those cells will become different type of cells in the brain of patients. But the fate of the cells is known, and at least is known in animals, and there is good control. There is no pure iPSC in those products, clearly. In our case, we work a lot with microtissues, and the same. Yeah, we check that we have no non-differentiated cells. So we still might have some progenitors of some kind, but they're already well advanced and they cannot go back to a real pluripotency, and that's very important in terms of safety. That's for sure. So that's a very, very important point. Then regulators, it's a new field, so there are some rules, but there are not that many rules that we can really put on the table. And I think safety is first. No teratoma, obviously, making sure that what you implant will not do more than what is supposed to do. I mean, put it very bluntly as a risk safety issue. Again, in regenerative medicine, it's very different from immunology, where cells are more transient, and I hand over to Greg on that.
Yeah, no, I think in the immuno-oncology space that we're in, or even if we're autoimmune, for example, I think you're right, the cell's probably more transient, but you still have to measure for residual iPSCs, and you need to make sure it's pretty darn low to non-existent. So I agree with everything you guys said about the safety implications of that. Maybe I'll just take it in a slightly different direction, if you don't mind. Is
Sure.
when you think about the advantage of iPSCs, like we said, is you can do multiple edits because of the proliferative potential. So then you got to think about the testing and safety of that. So if you say you're doing very precise editing, the FDA wants to know, and the other agencies probably want to know, what does the on-target editing look like? What's the frequency? How does it look? Is it just the piece that you wanted to put in? Did anything else go in? They definitely want to know about off-target editing, if there's any, where it goes. And I think it's unknown whether any off-target's okay, because think about lentiviral vector, it just inserts everywhere randomly. But precise editing, you pretty much want to be on target, and you can be, so that's good. So then it really boils down to genomic integrity. You want to prove that and stability. You want to prove that the insert is where it is and remains to be where it is as you go forward. So you make multiple batches. You want to make sure every batch really has, or maybe not measure every batch. Maybe in the beginning you do, but you want to have controls in place and confidence that every batch is going to make the same product with the same edits every time. And you're looking at various things. You're looking at chromosomal rearrangements. You're looking at insertions, deletions. As I mentioned earlier, integration of exogenous DNA other than just your payload, copy number variation, stuff that you can all imagine you want to look at. And I think because we have the ability to do this, and because we have a clonal population, at least in our product, and most of these iPSC derives can be clonal, you do want to do a pretty extensive examination. When you have a bulk population like primary derived, I'm not sure how you even do this. I think you might measure some things, but how would you ever know if there's some subpopulation that has something that's aberrant or deleterious? So with us, we, at least with iPSC derived, we can measure that clonal population, have a high level of confidence that what we measure is representative of every cell in there.
Got it. So just to clarify, so to what extent is, do regulators concern themselves with, the presence of non-IPS, non-target cell types? For example, you're trying to make NK cells, you get some T cells or something in the kind of differentiated within the differentiation pathway that is not fully functional, but might do something else.
I can start with that one. I think it goes, the answer is going to apply to any cell you make. So if you derive an NK from cord blood, in a previous life, I used placental cells and their natural immunomodulatory properties when I worked at Celgene. I think no matter what cell you make, there's going to be some impurities in there. So then it's just a matter of characterizing them, knowing what they are, doing the appropriate safety tests, potentially in vivo safety tests, certainly in vitro, quite a panel as well. And then you need consistency. If you're always going to have some small percentage of cells that don't have the right phenotype, you need to know what that phenotype is, and then set specs on that and say, if I'm always going to be below, I don't make it up 0.2% of whatever, I'm always below 0.2%. And that's back to process consistency. So if you can prove that, then you have a consistent, robust process and a reproducible product. Of course, making sure that the safety profile is sufficient, and of course, you still have the efficacy. When you have those impurities, then I think it's something that's acceptable.
Any further comments on that? Otherwise, we can move on.
Yeah, I fully agree with what Greg is saying. So it's the key word here is risk-based and consistency. It's a completely different story if your early step of the manufacturing process is not under control and you make some neurons while you want to make T cells. So even though if you understand that it might still be okay, but typically what we aim for is to have those early steps well under control. And so let's be honest, if we're talking about cells, if you look deep enough, and Frederick said this before, then you will always find differences. So this is not a small molecule where every molecule is looking almost the same. So cells are a little bit dynamic. Because of that dynamism, they actually work. And so the key is to understand that manufacturing process with in-process tests, but also with a robust QC package on the back end, where you characterize the potency of the cell, where you characterize the identity, where you characterize the impurities, and when those impurities are coming from slightly different cell types, then that's obviously, and when that is consistent. That's obviously compatible with a GMP manufacturing process.
I can bring in some more complexity around that. Here we're talking about like a pure product. I mean, a pure product. I mean, a single type of cell product. So being an NK or being a T or whatever. But when you start to work on macro tissues, if you want to do pancreatic islet, or if you want to do liver macro tissues or brain macro tissues, as I mentioned for Parkinson's, they are by construct. You have different type of cells, and they are not impurities, but you have to show that they are useful or that they are not harmful when they are harmless. And that's the name of the game. But there are situations now, we saw it in diabetes, where you create macro tissues which are not pure in terms of cells. But anyway, all the cells are called or named supporting cells. So no, it's not only impurities. It could be supporting cells for the life of the effective cells. So. So it's even more complex. So that's when we talk macro tissues again. It's a very different game.
Right. So that makes sense. I guess in the cell therapy space, we often hear the common refrain about how the process defines the product, and this is something you guys alluded to a little bit earlier. Again, I would imagine that in the iPS cell space, the different culture parameters that you could use could really impact the phenotype or the composition of the final cell products that you get, whether the particular cytokines or concentration of things, or the different steps there. So how do you go about testing the different variables, and also kind of how do you know what you're looking for at each of those steps?
Can I take that one? Well, just to start again.
Go ahead.
Yeah, it's a very good question. I think we have, that's development. We have to identify and to quantify and to understand what are the critical parameters of the protocol we're using. Otherwise, let's say the reliability of the process is not there, because small changes might have a large impact. So it could be a change, as you said, in concentration of some factors, could be the timing, could be the combination of factors, could be a lot of things. So there is a lot of work, and that's a specificity of cell therapy. There is a lot of work before you even go into your tox study in animals, because you have to go in your tox studies with your final product. So, which is very different from even antibodies. Antibodies, you can go with I would so-called a dirty antibody into animals, because after that you will purify it more and more, and that's okay. People understand it. It can be worse. But in the case of cell therapy, it doesn't work like that. You have to have your final protocol, and that what is an issue for the sector. It's when usually you go to tox studies in animal, you go there for proof of concept study. So you have a limited amount of materials, and you're doing it, let's say, at the bench. And even for tox studies, most of the people don't go to a CDMO or GMP facility. So then you get your tox results with the product, and then when you go to human, you have to move to GMP to transfer your technology to a GMP site or to GMP facility, and maybe the product will not be the same. So maybe the QC will be the same. So you will release the same product, but there will always be a question mark on, is it exactly the same product because you've changed this or that? So if you know well your critical parameters, you can change some others, and you can say, okay, this is a minor change, and this is not impacting. Your QC is the same. The product at the end is the same for everything I know about it. It's the same. I have changed those little parameters, and they were not critical in my protocol. So, that's okay, and that's a minor change. It's okay. But if you touch a critical aspect of it, it's much more complex.
Good question. The process is the product. Unfortunately, yes, the process does impact the product. In fact, the way I think about it, it's the gene edits is driving potency for a large extent, but that's really in combination with the manufacturing process. So those two together form the quality attributes of the drug product that is manufactured at the end of the day, and based on that, QC criteria need to be set. So typically with iPS, so I mentioned before, these are complex manufacturing processes, and when you change basal things, you see surprising changes. It's well documented, for example, in autologous NK manufacturing that cells are different pre-expansion and post-expansion. That is true in the iPSC context as well. So how to set it is, you want to understand the target product profile that you're working towards, and based on that, you develop analytical methods that are measuring those key attributes, and from there on, you try to focus your development teams on achieving those goals. Ahead of that, there are different developmental steps, and typically what you try to do is to be inspired by developmental biology and follow the normal developmental paradigms, because that well gives you confidence that you're making the right cell. And so, but at the end of the day, it's a lot of hard work. We're testing a lot of variables, and that is, I think, one of the interesting things.
Yeah, if I could just add, I think it's an ongoing process to look at all these variables. So as Frederick alluded to, when you first make your tox material for those first studies which support your IND, you have a certain framework of a process. I think it's important to know your process can be consistent. I think it'd be dangerous to just run it for the first time ever and say, okay, I made my tox material, because who knows if you can reproduce that product the same way. So you want to have some characterization, but let's be realistic. You can't characterize every parameter before a phase 1 study. It's just not going to happen. So you get better and better as you go along. So back to Frederick's point, you definitely have to have some sort of, maybe confidently is a strong word, but you need to be confident that the material you put into animals is representative of what you're going to later put into your phase 1 studies. You need to show some analysis of those materials, maybe just by release, maybe a little more than that. But as you go down the path and eventually get to a BLA, then you ultimately process validation, which says, I know what critical parameters look like. I know what the ranges on them need to be to meet my critical quality attributes, and I validate all that and prove that I can make the same product every time if I control parameters within a certain range. But in between, you just constantly learn. And that's what people talk about when they say quality by design. You're constantly trying to understand which parameters, input parameters, affect which outputs, and then you get a design space. So it really becomes a matter of how much you want to invest early. You could use things like risk-based analysis to look at your and we do this all the time. You look at your overall process and say, there's dozens of steps, maybe hundreds of steps, hundreds of parameters. Dozens of steps, hundreds of parameters, let's say. And I can't measure every one, so you do a risk-based approach. Which ones are the most important? How do I prioritize experiments? Ultimately, you do more of them. You use design of experiment to look at multi-parameter interactions, and then eventually put a validation package together. But you have to realize the validation bar for a BLA is much higher than the process understanding needed for phase 1. It's finding the right balance along the way.
Great. So we've talked a lot about different elements of iPSC manufacturing. What do you all see as the biggest challenges with respect to iPSC manufacturing today? Or put it another way, are there issues that or challenges that you think don't get enough attention? Or there are no challenges and everything is perfect?
And I'm going to speak out of turn here. This will probably help me on this, but the science itself, the differentiating an iPSC into the cell you want. And I think my colleagues on the line have more expertise on that than I do, just because I'm more of an engineer that's designed processes. I don't design the process and scale up. I think there's certainly people on the research end that understand the different protocols and why we add which cytokines we add when, and ligands when to stimulate what, to go down the NK pathway, the T cell pathway, or others go down the cardiomyocyte or the neuron pathways. I think being able to understand all that and control that properly is probably the greatest challenge, because at the end you need a functional cell. I think the other big challenge that many people are pursuing, not all, but is the gene editing piece, because as I said before, gene editing, there's no exact formula to know exactly how you're going to want to do those, let's say 3 sequential edits you want to do, and how they are all going to work out. And you're going to get the cells to not only express the CAR you want to express that you edited on, but they're still going to have good function. So being able to do all that and have the the on-target editing you want, no off-target, the genomic integrity and stability, it's not easy to do. I'll tell you right now, there's a lot of starts and stops along the way. So having strong expertise in that, and then understanding what to measure once you do that. It's not just, like I said, just expressing the car. It's, does the car work? Do the cells still work? So I think Stefan alluded to this a couple of times. It's a really, really complex technology. That's the power of it, but you better have a lot of expertise to be able to do it well.
Yeah, so I think, so looking backwards, maybe 20 years ago, so a lot of people started to work on cardiomyocytes because they were easy to identify. So you saw some beating cells in a dish, and so at least you had a clue what you were doing. That was the honest truth. And so looking where we are right now, so the hardest problem is to instruct the cell to become the cell type that we want to make. And so we are finally at a point that this becomes robust, reproducible, scalable in bioreactors. So we are not talking about a few tens of cells anymore, but billions of cells. So I think it's not 100% solved, but multiple players in the industry have shown that they can do it. So what I think right now is a big open debate is the amount of gene edits needed to make a therapeutic relevant cell. We haven't really talked about this, but we and others are very focused on allo. To make an allo cell therapy, there are certain genes that do not help you, that might give graft versus host. They might lead to immediate rejection of the graft. So knockouts are needed. You see that in Greg's pipeline. But I think the thing that people cannot answer today yet is what that package of edits is looking like, what you really need, how that impacts the safety of the product. So it's going to be, and there are no good animal models out there right now. So the early clinical trials are going to be really interesting to see what is that package that is needed to show efficacy in the various indications that are being pursued.
I think, again, with the angle of regenerative medicine, again, which I think is a bit different from the immuno-oncology market approach. We will need a lot of cells if we want to be successful. If you take a disease like acute liver failure, you will need a few billions of hepatocytes per patient. And if you want just to run a batch of let's say 1,000 patients, which would be let's say a regular batch. It's not a huge batch, it's just a regular batch. It means that you will have to make trillions of cells, trillions of effective, alive cells. And then you can freeze them, store them. So today, people can, if you go to a company that produce iPSCs or cells, they usually can handle a few hundreds of samples at a time, because it's such a work to freeze cells. Just freezing cells is difficult. So when we want to do that at a larger scale, like 1,000 doses or 10,000 doses, we don't know how to do it yet. I mean, with the regular, let's say, control of the pharma industry. So there are a lot of challenges like that because we are changing scale. And just to give you an example, last year we published what we thought was the first bioreactor of iPSCs of 10 liter with 100% survival. Okay, so like this was very good. We had something between 15 and 20 billions of cells in this bioreactor. We had to throw them away because we didn't know how to handle that many cells. 15 billions of iPSCs, that's a lot of value in the market, as an iPSC cell, it's a lot of value. But there is no way to handle such an amount of cells. So that's something we are working on, really. Also the downstream processing of those quantities we will need. So not only we don't know how to produce them, but we don't know how to handle them when we produce them. And again, it's very different in the immuno-oncology sector, where cells are, single cells in suspension, and very different from what's happening in the RegMed segment. So a lot of even engineering challenges still to be covered.
I'll just ask one quick question in the last minute or so that we have, which is, or a couple of minutes that we have, because we have a little flexibility there. What is an important innovation that you're excited about that could shape manufacturing for the space in the coming years?
I need to pass that one because I think the answer is here, but
Yeah.
I will let Stefan saying it, so I'm not saying it.
Stefan?
I'm not going to talk about tree frogs. So, no, so I'm super excited about the fact that we are what I just said, that we are starting to see batch sizes in the iPSC field on billions of cells. So we have produced cardiomyocytes at 30-40 billion liter scale, and so that wasn't pushing the process. We are on the right path to do the similar things with NK cells, with T cells. We have done it with endothelial cells. We have scaled lung progenitors to the billion scale. So we have done it across a number of different cell types, and so I think the proof points that you actually can manufacture these cells are now out there, and so this is the right time to make the cell therapy a reality. And so and for me, so looking at the CAR T field, so it's amazing what these therapies are doing. But if you see the factories that therapeutic developers have to build, and if you then look at the output of such a facility, it might actually be only 5,000 patients a year. And so the science there doesn't match the demand from the clients, from the patient side, and cannot be paid for by the healthcare system. So new technology is needed, and iPSC technology fits there in that pocket and is there to revolutionize these treatments.
Yeah, I'll just add on to that. Stefan's right. You know, these autologous therapies are great. They really help patients, but not only is the cost really high, but they can't make them fast enough. They can't get patients accommodated fast enough because these facilities that are so big just can't handle enough patients. So patient access, because the product's not available or the product's not affordable, is a big problem now. So that's where these therapies come in, is that we can make them cheap and make large amounts of them. And that's going to help so many more patients.
Great. In that case, I see we're just at the end of the session. So I will thank each of you again for the participation and the fantastic discussion.
Thanks for having me.
Thank you for having us.
Thank you.